This is iGEM Team again. They will have 20 minutes for presentations, followed by 5 minutes Q&A. Let's begin.
Okay, so let's go, and welcome to our iGEM project presentation.
My name is Lisa, and this is Yannick, and together we represent the iGEM Team Göttingen.
In our project of this year, we dealt with one of the most prominent herbicides all around the world, namely the glyphosate, where we're trying to find a bacterial Roundup approach to the glyphosate controversy.
So we wanted to find a bacteria-based method to detect the weed killer that can be used by everyone at home without expensive laboratory equipment. And on our way to find a suitable topic, we went to different panel discussions, we looked at the media, news, and the one topic that continuously popped up was glyphosate, making headlines all over the world, ranging from Europe, over Asia, to the United States, just underlining how important the topic is everywhere.
Now, if we take a look at the triumphal profession of glyphosate, we can see that ever since it has been patented in 1974 by Monsanto, the usage rate of this herbicide has been ever increasing to this very day and is still on the rise. Glyphosate is a broad-range herbicide and the main component of the Roundup products and is sprayed to the fields. Over the past few decades, this even has caused some natural resistances to occur, specifically in the countries where it's most used, like the US or Australia.
So as I already mentioned, we talked to a lot of people, and even though the majority had heard the name and the alleged problems that came with it, only a few understood the mode of action. So how does glyphosate work?
Oops, sorry. Now, if we take a look at the molecule of glyphosate here to the right, we see that glyphosate inhibits the shiggy-mate pathway in bacteria, fungi, and also in plants. A certain synthase of the shiggy-mate pathway is inhibited through the glyphosate, so the active center is blocked, and in the end, some essential aromatic amino acids can't be formed. But instead, we observe a pathway interruption right here, causing, for example, a plant to wilt or bacteria to die, as the organism that is exposed to glyphosate starves for these essential amino acids.
So glyphosate is the most controversially discussed chemical at the moment, and we created a survey asking a broad range of different people on their opinion on glyphosate, including students, faculty members, common citizens, and politicians.
Of these people we asked, around 25% favor the usage of glyphosate, as of course some big benefits come with the use of the weed killer, and it is also a cheap and reliable herbicide that can be used over the whole year.
On the other hand, 75% had a negative view on glyphosate, which is mainly due to the fact that it was recently detected in food items, but also the major concerns were its accumulation in the soil and with that the effects on biodiversity. However, this is not the only problem. Another big problem is the controversy in science, where huge agencies such as the World Health Organization or the European Food Safety Authority contradict each other. But one of the most recently published paper, which was just published like a couple of weeks ago, talks about the effects of glyphosate on the gut microbiota in honeybees, which leads to more infections or more pathogens in the bees, finally leading to death of the insects all over the world, linking glyphosate to another important problem of the modern world.
Also, just recently, there has been a lawsuit against Monsanto, as this guy right here, Dwayne Johnson, sued this big company to be responsible for the cancer development. So you see, all in all, glyphosate is a really controversial and diverse topic.
So as I already mentioned, our main goal was to find detection methods to detect glyphosate. So for this, we first had to analyze the interaction of bacteria and glyphosate. We needed to create resistant bacteria, which we can further use in the detection system, and we wanted finally to produce a product. So we needed to find a suitable bacterium. And the spore-forming soil bacterium, Bacillus subtilis, was the bacterium of choice.
And if we take a deeper look into the traits of Bacillus subtilis, we quickly understand why it is so great for our experiments. It is a soil bacterium. Being naturally present in the soil is a big advantage, as glyphosate is also applied in the soil. Why being present there, it also has been reported to enhance plant growth. And also, Bacillus is a non-pathogen that even competes with plant pathogens itself.
But it's not only beneficial to plants, it was also shown to improve the health and digestion of livestock. It's also known as the industrial workers, being the model organism coming right after E. coli. However, even though it possesses all those characteristics, the interaction with glyphosate is poorly understood. So this is where our research starts.
And of course, we first had to take a look now. How would Bacillus react if it is exposed to glyphosate? And we did just that, exposing Bacillus subtilis to low amounts of glyphosate, ranging from 0 millimolar up to 3.5 millimolar of glyphosate. As a reference, a normal Roundup bottle that can be purchased in your local store contains around 40 millimolars of glyphosate. And so we already see on this graph that the bacterial growth is severely influenced by the glyphosate presence, and we observe a big cell death occurring. So we knew we had to strengthen Bacillus in the process to go on with our experiments, asking ourselves the question, can we make Bacillus great again?
So for this we used adaptive evolution. So we've used the bacteria and basically cultivated them in increasing glyphosate concentrations. And you can see that at low glyphosate concentration they grow in a bacterial lawn, and you can also see a reduced growth with increasing glyphosate concentration. And at even higher glyphosate concentration, suppressor mutants rapidly emerge. But we wanted to make them even more resistant. So we picked single colonies from the suppressor mutants and repeated the experiments, so that again you can see at low glyphosate concentration a bacterial lawn, and with increasing glyphosate concentration, suppressor mutants. And those suppressor mutants were even resistant to up to 50 millimolar glyphosate. And those early experiments already showed us that Bacillus rapidly adapts to glyphosate concentrations, which is very good for us because this means that we can use it for further experiments.
But in order to do so, we first have to understand why Bacillus is even able to acquire such resistances. And to do just that, we performed a whole genome sequencing with the help of the genomics lab in Göttingen. And we observed a lot of mutants that we picked from our plates. And there we discovered that in every mutant that has acquired some sort of resistance, we observed deletions in a certain GLTT gene. And as the resistance is higher with increasing glyphosate concentrations, also some changes in the GLTP gene of Bacillus subtilis, rendering those two genes as defect or inactive. And if we take a look at the function of these genes, we realize that both of them code for glutamate transporters in Bacillus subtilis. However, now through our experiments, we could discover that GLTT and GLTP are also involved in the glyphosate transport for Bacillus subtilis, which makes them the first glyphosate transporters ever known to the science community. And we are very proud of this breakthrough in our project.
And you also have to consider that glyphosate exists for over 50 years, and those are the first transporters identified. Because those transporters are the foundation for all our further experiments, we picked it to be our best part. And it's not only the major entry for glyphosate, but it also has many biotechnological applications. And one huge biotech company already contacted us to use the resistant strain to detect additional glyphosate transporters in plants.
Now that we understood the whole background of glyphosate and Bacillus subtilis and acquired resistant bacteria in the process, we come back to our initial aim, which was the development of a functioning detection system with our bacteria. We therefore took upon two different approaches, one of which being the enzymatic detection and the other one dealing with fluorescence proteins. Both of these methods take interspecies competition as their basic concept.
So for this we used two strains, a resistant strain, which is shown here in yellow, and a sensitive strain, which is shown here in green. And we co-cultivate them in the same ratio and let them grow at increasing glyphosate concentration. So at low glyphosate, they should grow in approximately the same ratio, while at increasing glyphosate, the sensitive strain will die, while the resistant strain will persist. And because this is the base of our experiments, we wanted to model this. So we took the growth rate of the 2 single strains in presence of glyphosate, which then allows us to model the whole competition assay. And you can see that at low glyphosate concentration, the sensitive strain even has a slight growth advantage in comparison to the resistant strain. However, with increasing glyphosate concentration, the ratio changes in favor of the resistant strain. And we could already use this to determine the glyphosate concentration of a soil sample. However, we can't say for sure that the sensitive strain dies because of glyphosate or maybe because of something else in the soil sample. So we need to put them in competition. And we want to visualize that the sensitive strain dies and the resistant strain survives. So for that, we use fluorescence proteins. And here we have, on the one hand, the resistant strain, which is tagged with an orange protein, and on the other hand, the sensitive strain, which is tagged with a green fluorescence protein. Both of those fluorescence proteins were obtained from this year's BioBrick kit provided by iGEM. And then they do what I already explained before. So we let them grow in a 1-to-1 ratio and let them fight for their lives. And this is what happens. At low glyphosate concentration, they grow at approximately the same ratio. However, with increasing glyphosate concentration, the sensitive green cells die, while the yellow cells persist. So that at the end, almost only yellow cells are left. And when we take an even closer look, you can see that at low glyphosate concentration, the sensitive strain grows very well, and therefore also the fluorescence intensity is really high. While with increasing glyphosate concentration, the cells die, and therefore also the fluorescence intensity decreases. And in contrast to that, you have the resistant strain, where the fluorescence intensity is rather low at low glyphosate concentration and high at high glyphosate concentration.
Now, at this point, we already were able to establish a completely novel detection method for the analysis of toxic substances, as this method could not only be used for glyphosate, but be adapted to other toxic substances as well. Still, we need some pricey lab equipment to make this possible, so we took upon the other challenge, which is the enzymatic detection.
So this is based again on interspecies competition. However, this time our resistant strain is not yellow, but colorless, so it has its usual whitish color. And on the other hand, we have the sensitive strain, which contains the laxet gene. So in the presence of the substrate IXGAR, the cells turn blue. And then we do what we already did before. We let them fight for their lives.
Now, what we get out of this can be seen here. As you see, a nice color curve ranging from the more bluish color up to a white color over here. And this can be explained as the sensitive strain has the blue color and has a natural advantage over the resistant strain if no glyphosate is present. However, if we increase the glyphosate concentration, we observe the color change to more whitish as the resistant strain claims its advantage here. The whole detection can be compared to a pH indicator paper. So very basic system that everyone can understand. And we also quantified this approach through the use of a beta-galactosidase assay. As you can see, the enzyme activity decreases while the glyphosate concentration increases. All in all, we can say that we have an anti-proportional relation right here, where the glyphosate concentration causes a color switch in a way that everyone can read out how much glyphosate would be present in a sample. And we took this idea and realized it in a prototype that now our little big Sherlock subject will present to you. As you can see here, a product provided by our sponsor Sartorius enables the application of some water and soil mixture that was contaminated with glyphosate to the top of a filter tube. And now, if you apply it right there, some spores of our bacteria together with dry medium will carry out their detection reaction, leaving this color curve behind that I just spoke about. So now we close it. In the end, we would see the color curve if we apply some samples. Thank you. Now, at this point, we already were able to successfully complete all of our initial goals of the project. So as you can see, everything is done till now, but this wouldn't be iGEM if we weren't up to something more, because on our way through the project, and as you all know, an iGEM project is a big process, we got in touch with the public, took part on panel discussions, organized one by ourselves, visited schools and held many presentations. And one question that always popped up was, what can be done about the inactivation of the weed killer? And are all bacteria potentially able to not only detect them, but also maybe inactivate them?
So we use Roundup or glyphosate to kill our weed, and after the weed is dead, we use our engineered bacteria to inactivate glyphosate to clean up contaminated sites and fields. And for this, we need to arm our bacteria for its next battle. So we use glyphosate N as a transferase, or short GATT, which originally came from a close relative from Bacillus subtilis. And it's also commonly used in genetically modified plants that are resistant to glyphosate. The gene was provided by Integrated DNA Technologies, which was free of charge because they collaborated with the iGEM competition. So we engineered our bacteria in the way that it has the synthetic gene. And in presence of glyphosate, it acetylates glyphosate, leading to an inactive form.
Now, if we visualize this once again in the graph, we can see that for the wild type, at around a concentration of only 1 millimolar of glyphosate, the growth rate is at its half maximum, while the gut-engineered strain possesses an around 10-fold increased resistance to the glyphosate, so that these bacteria most likely take up the environmental glyphosate instead of avoiding it, and also potentially inactivate the weed killer in the process. So a success also for our inactivation experiments. Now that we are at the end of our presentations, we want to summarize all of the achievements that we collected through our way of the iGEM project. Starting off, of course, with a big finding, which were the 2 transporters in Bacillus subtilis that we used for our ongoing experiments.
We also did not only find 1, but 2 novel detection methods. The first one is based on fluorescence proteins and is very precise. However, we need laboratory equipment. The second one can be used by everyone at home without expensive equipment.
We therefore also designed a prototype of a product that can be used by everyone, that uses this system that can be understood by everyone.
And finally, we can not only detect it, but we can also then inactivate it and clean up contaminated sites and fields.
Of course, an iGEM project is a big challenge, I should say, and we had some help on our way, so we want to acknowledge some people at the end of our presentation. Starting off with our supervisors, Fabian and also Jörg, who both have been a big inspiration throughout the whole project and took our team through ups and downs. Also, a big thank you to our sponsors who made the whole project possible.
We are also very grateful to Dr. Stefan Plum, who helped us with the modeling, and to the iGEM team Marvel, who collaborated with us and carried out ITC analysis.
Now, at the end, we of course want to thank you all for attending our presentation. Maybe you can also take a look at our poster, but for now, thank you and have a nice time at the ongoing Giant Jamboree.